Parkinson disease is a progressive neurodegenerative disorder that initially affects the substantia nigra in the nigrostriatal pathway, but later it also spread to other areas of the brain. It is characterized by altered motility, but also there are non-motor symptoms such as lower urinary bladder tract symptoms. The prevalence of LUTs in Parkinson disease is up to 85%, and irritative symptoms are more frequent, such as nocturia, frequency, and urgency. It is correlated with disease severity and disability, and it has a huge impact in the quality of life of these patients. When checking the neural control of the urinary function, I figured out this is a really complex process, and still is not fully elucidated. But what we know is that the substantia nigra and the VTA play an important role in regulating voiding through the PMC and the autonomous nervous system. The 6-hydroxydopamine rat model of Parkinson disease is a well-established model to use in this case. The 6-hydroxydopamine is a structural analog of dopamine and acts in degenerates the dopamine neurons by causing oxidative stress and apoptosis. It has to be injected directly in the brain since it cannot cross the blood-brain barrier, and can be injected in different areas in the nigrostriatal pathway, as you can see here, creating different lesions, in this case a partial lesion, and in this case a total lesion of this pathway. Previous preclinical studies using the 6-hydroxy rat model of Parkinson disease showed signs of the trusor overactivity similar as we see in Parkinson patients. For instance, a decreased bladder capacity, as shown here, are a higher frequency of micturations, as you can see here. Our research group also previously showed that there is a local plasticity of the urinary bladder contractility. When the tissue is stimulated electrically, there is enhanced contractility, and also when it's stimulated with cholinergic agonist, there is also enhancing the contractility of the urinary bladder. So knowing this, we wanted to further investigate if the substantia nigra or the VTA or both are involved in the altered urinary bladder contractility seen in the 6-hydroxydopamine rat model of Parkinson disease. We use Sprague Dolly animals, and we divide them in 3 experimental groups: controls and 2 6-hydroxy variations of the model, MFB lesion, where we lesion the MFB area, and the intrastriatal lesion when we inject the neurotoxin at the striatum level. The animals were adapted to laboratory conditions for a week and were followed for 4 weeks after the lesion in the brain. Then they were sacrificed and the tissues were collected. For the urinary bladder, we perform isolated tissue baths, like as you can see here, and we stimulate the tissue electrically with a cholinergic agonist and with a purinergic agonist. And we confirm for the deletions with a thyroxine hydroxylate staining. Here we have a control. Here we have a 6-hydroxy intrastriatal lesion when the VTA is almost intact. And here we have a 6-hydroxy MFB lesion when the substantia nigra, the striatum, and almost the whole VTA is gone. What we saw? We saw that First of all, here we have contractile response, and here we have the stimulation. The blue line and dots and squares are the MFP group, the red one is the intrastriatal, and the black ones are the normals. What we saw is that it's an enhanced contractile response in both 6-hydroxy groups when they were electrically stimulated. It was a little bit different when we start stimulating the tissue with cholinergic agonist, when only the 6-hydroxy MFP group was enhanced. There is a slightly trend in the 6-hydroxy intrastriatal as well, but not so clear. And when we stimulate the tissue with the purinergic agonist ATP, then we only found a trend to increase in the 6-hydroxy MFP group. Meanwhile, the 6-hydroxy intrastriatal group was without change. So how we can interpret these results? First, we can argue that might be a change in the receptor expression at the peripheral level. This means that the muscarinic receptors and or the purinergic receptors are upregulated, that might be. Or the enhanced contraction that we can see is cholinergic, pure cholinergic effect, or can be just a sympathetic or adrenergic effect because there is a decrease in the inhibition of the contraction. Or the other is that since the tissue has to contract many times, that can produce also local inflammation and then lead to a hypertrophic small muscle. Or maybe a combination of all of them. To conclude, from the electrical stimulation data, the Substantia nigra and not the PTA is likely responsible for the local changes seen in the true storm contractility. And from cholinergic and purinergic stimulation data, we can say that impairment of the BTA might further aggravate the urinary bladder dysfunction. As a take-home message, both the Substantia nigra and the BTA might be involved in the urinary bladder dysfunction seen in patients with Parkinson disease as the disease progresses. Thank you very much.
Thank you very much for this nice presentation. It's not now open for questions. Naoki?
I'm Yoshimura in Pittsburgh. Thank you for showing me my results in your slide presentation. So what the timing of these changes occur? So you saw that 5 weeks after the 6-hydroxydopamine injection? Your study design is
Yes.
5 weeks after. So how early this type of changes is found? So even 1 week or 2 weeks after the injection, you see these changes?
We did not stop
time course of the changes.
We did not study the progression of the lesion before the 4 weeks, but in previous studies has been shown that the degeneration gets after 3 days, and then is stable after 14 days and up to 28 days. So that's why we decide at 4 weeks to make the analysis when the whole lesion was stable.
Okay. So then also that 2 different injections of 6-hydrodopamine shows a similar level of reduction of dopamine in the brain. Have you ever looked at the dopamine levels in the brain after the 2 different treatment?
Yeah, that is an interesting comment actually, because we did not really mention directly.
Yeah, so MFB targeting is more very precise tract, but an intracellular injection is kind of broad. So sometimes not effective to reduce the dopamine level. That's what we found. So that's why we are injecting Substantia nigra directly. So that was a 6-hydrodopamine injection. So I saw that a technical problem for induce that dopamine deletion in the brain region. That's what
actually
probably
true.
You have to confirm that.
Yeah, and that's actually true because then we also found the degrees of the lesion, right?
Yeah, yeah.
So you can
degrees of
have
degrees.
50, 60, 75, 80.
Yeah.
So maybe that is
Maybe.
interesting to see the correlation between that
That's may
dopamine loss.
related to the difference in the
Yes,
bladder
exactly.
reaction.
Thank
Martin Michel from Mainz, Germany. I have a question that relates actually to Naoki's last question. The mouse brain is small, so missing the spot of injection or injecting a dose that spills over into areas other than the specific spot you wanted to target can happen easily. Did you verify that your injection really was where it was supposed to be and that the effects were limited to that region?
Yes, we need to confirm the lesions to validate the data. And when to do so, we actually do this staining. So we can see the degree and do some densitometry measure to have a sort of a objective way of dividing these two groups, right? But if I can tell you, when we were doing the experiment, at the beginning, since I was training as well, the intrastriatal group started to be a little bit too lesion. So it started to be so similar to the MFP that we actually checked what was happening. So we use methylene blue to actually check where the exhale injection was delivered. And then we found that we needed to do an adjustment to the experimental design of the lesion of the intrastriatal to avoid that circumstance you mentioned.
Well, I have a question. Did you have any observation of the animals during this development suggesting that they have, for example, the true sphincter dysynergia? Because if you have that, that could be an explanation why you get the changes in the muscle, because you could have an increased pressure and hypertrophy. So your speculations are most probably correct, that you had some kind of hypertrophy and that would increase the contractility of the muscle. But you don't have any in vivo observations that could correlate to the changes. Have you done that, or?
No, that is the next step for us. We will start actually using these models in an in vivo setting to explore a little bit more how actually happened physiologically in this model.
We have published a paper, a couple of this monkey model and also rat model, but we didn't see much in change in urethral function like a DSD type of obstruction. Just overactivity then can void almost completely each time.
You didn't see DSD?
No.
Well, we did.
You did?
Yeah, well, we were cited in the study.
Okay. Okay. So my case, we didn't see much, just as they have just overactivity, no obstruction.
But we also looked at the bladder size, and there was hypertrophy in the muscle. And we assumed that this was due to the DSD.
So maybe difference in the duration of the disease condition, maybe. That's probably. Okay.
Could be,
yeah.
Did you weigh the bladders?
Yes.
Yeah. How much did they increase in weight?
That is the thing. There was no difference in the weight, but it was more difference in the texture. When you remove them from the animal, the 6-hydroxy groups has a slightly thicker bladder, but it was not changing in the weight.
So one would suspect that if they had the true sphincter dysynergia, the bladder weight would increase. Yeah. Yeah. Okay. Yeah.
Well, oh yeah.
Yeah.
interested in your in vitro experiments.
Thank
Could you just show the slides again, please, for me? Just that one and yes. So you saw an effect at high frequencies, but not low frequencies. And several people have shown that at low frequencies, ATP is a predominant neurotransmitter, whereas at higher frequencies, acetylcholine is a transmitter. So it's not in disagreement with your results that when you put the agonist on, no effect of ATP, but a small effect of acetylcholine. Did you try to add atropine to these tissues or to desensitize the purinated receptors, and then you would see perhaps a clearer result? Have you tried that or not?
Yes, we actually add atropine.
Mm-hmm.
So in the presence of atropine, we also made the same curves for methacholine, and also the presence of atropine for ATP.
Okay, so you would expect to see no effect if you added atropine, and a big effect if you added something that would desensitize purinergic receptors.
You can see a decrease in the contraction, although not abolished completely when you add atropine.
Okay. I mean, the other point to make of this discussion about hypertrophy, if you normalized your tensions to grams of tissue weight, then you don't need to argue because you've normalized the data. Very So just.
Thank you for your observation. Thank you very much.
Thank you very much.